You can hear me, right? Yeah, all good. Yes. Flash recaps, day 4. This page. Quickly do all the labeling. Anything you don't know, any labeling you don't know. Inferior vena cava you have, superior vena cava you have. Label the 4 chambers, 4 pulmonary veins, one on each side, bringing blood to the left atrium. Then pulmonary artery dividing into 2. You don't, here you can divide the pulmonary artery into left and right. Right? Divide the pulmonary artery into left and right. And what else? This is the aortic arch. Label it. And label the muscles, papillary muscles. And the cords suspending. Use my paper. Chordae tendinae. Right? Chordae tendinae. Okay. Do the labeling quickly. Okay, I'm sending online people. I'm sending the flash recap PDF for now. You will receive delivery of it once you register at the website with the address. Right, it will be delivered to you. For now, refer the PDF for today. Okay, do the labeling quickly. I sent to the group. Okay, quickly refer it. And okay, I want you to write down some points underneath. ඉඩ තියෙනවනේ දෙයට. Right, some points. Write down underneath. මෙහෙම right, heart wall has 3 layers, right? After doing the labeling puta, like write down small points, right? It's just a short note, part of, it's not today's lesson. We are just doing it. It's a revision class, right? Heart wall, what are the 3 layers of the heart wall? Tag gala kiyan pericardium is the outermost layer, මැද muscular layer, myocardium. So today you have to go home and revise what we did in the flash recap, right? I'm going to go through these lessons, but we have done, right? Myocardium, pericardium, and innermost එකනට අපි කියන්නේ මොකද්ද? Endocardium. The heart wall has 3 layers. So write down the 3 layers. Okay. Be careful about the which side has the bicuspids, which side has the tricuspids. Chordae tendinae, pay attention. Chordae tendinae, draw on your books, because they are not in the diagram, right? Chordae tendinae and the papillary muscles, they are not in the diagrams I have included, but they are in your syllabus. Yeah, I forgot. Semilunar valves. Where do you have the semilunar valves? Here, no? Aorta, where the aorta leaves the heart, and where the pulmonary artery leaves the heart, you have what? Semilunar valves. Why do we call them semilunar? If you take a look, they look like 3 half moons, right? Half moons, right? So Semilunar valves can be found where in the aorta and where the pulmonary artery starts from where? Right ventricle. No? Yeah, that's all. You are at freedom to write whatever the things you think you want, right? In the remaining space. This is all I'm giving. Huh? Online people, anyone who's not having the genetics and molecular tute today, PDF, even the PDF, hands up, send me a message. All of you have. Didn't receive or what happened? Send me a message. Uh, you didn't receive the delivery of it or nothing. I'll send the PDF to the group. Okay, I'll look into why you haven't received the tute. Okay, I'll ask. All right. Refer the PDF for today. Okay. All right. So this is not our main focus today. We are going to start molecular today. Right. Molecular biology. Uh, all done, right? This is fine. These are simple things. Okay. You can. I mean, you can do a nice self revision by yourself. Heart structure. These are basic things you knew from O level days, but some additional things you need to know are mainly the papillary muscles and the three layers of the heart wall. That's it, right? O levels filling, nothing much added in the terms of heart structure. Okay, online people all good. Okay, right. So today the plan is to move on to molecular, but we have a small part of genetics left. Right, we have a small part of genetics left to discuss. That is breeding techniques. No, we are in breeding techniques. Up to where did we talk about breeding techniques? Breeding techniques, that's a very, say, let's say, common source of MCQs, right? Breeding techniques. Come to page number 123. So we started with artificial breeding, artificial selection. Okay, I think we have to continue today. Outbreeding or inbreeding. We finished inbreeding. Right before that. So what is actually breeding? We have talked about so many genetic techniques. right? Genetic techniques. But these are real life applications of these genetic techniques, right? You don't bother to study these things, right? Because very boring, just theoretical things. But if you take past papers, there are MCQs from different types of breeding techniques, right? So page number 123, resource book, grade 13 one, yes. Yeah, sorry. Yeah, it's not in the grade 12. Unit 6 is in grade 13. Yeah. All right. Can you share? Okay. Right. So, we are making a small note as well. For genetics, we made a small note. Do you have it completed? Not much. No? For you, I have the recordings. Okay. Genetics, we started in December, right? December first week, we started genetics, right? Yeah. December second week or first week, we started. And December last week was a holiday. No? So, this is our day 4 genetics, right? So not much. I took some time doing some questions. That's why it took some time to discuss genetics. Right. Anyway, so we have come to these applications of genetics, breeding techniques. Right. So we started with the simplest one. If we come to breeding techniques, artificial selection. Right. I'll quickly summarize what is artificial selection is. That is the oldest form. So two things you need to do. First thing, highlight the resource if you haven't. Resource එක highlight කරගන්න. That means you have gone through it once. That's the first step. Then take down whatever you want in the form of a short note. මං කියන්නම් ඕනේ like take this down, or you can take whatever you want, right? What I write in the book, right? So artificial selection, it has few steps, right? What is a it's a person who has just some general knowledge can do artificial selection of breeding, right? As a very simple farmer, ඉස්කෝලේ ගිහපු නැති ඕනම a simple farmer can do artificial selection, and they do, right? What they do is plants and animals with specific nice traits, right? For example, if you take let's say fruit plant, the biggest fruits is the desirable trait. ලොකුම ගෙඩි හැදෙන එක is the trait you want. So you choose it as your trait, and then you only allow the plants producing the biggest fruits to reproduce. The fruits producing small, sorry, the plants producing small fruits are not allowed to reproduce. You are selecting who gets to reproduce. Very simple. So what's the definition? Process of selective breeding where plants and animals with specific traits are selected to breed so that their desired traits could be passed to the next generation. So it's a stepwise process. To do artificial selection, I think I talked about this last week. To do artificial selection, the first thing you have to have is variation. You have to have a variation. What does that mean? Let's say you have a fruit, pumpkin patch pumpkin make right? Sort of cultivation of pumpkin. So within that pumpkin cultivation, there are going to be small pumpkins and big ones. Variation. So what is the nice trait that you want to pass is the large size. So first thing you have to have is variation. Then what you're going to do is you are going to select best performing individuals. Neither select the best performing individuals. Then what you are going to do? You are going to interbreed them. Only the best performing ones are allowed to breed. Then you get a new population, and that population you are going to screen. In that population also there can be big ones and very big ones. Neither. because now you have sort of taken one step. So in this generation, what are you going to do? You are going to still again choose for the biggest. Then you are going to again interbreed them until you keep on breeding until you get what we call as a new variety. Until you get a new uniform variety. This is the end target of artificial selection. Okay, simple. We talked about these things. Today, we have to move on to something called outbreeding. So, this variety is something you need to know before we move on to outbreeding. Page number 125, outbreeding. Okay, okay. Inbreeding we discussed last week. I'll send you recordings. Okay, are you in the groups? In the group. Everyone is in the group. Online people, you can hear me all good, right? Yeah. Outbreeding, as the name says, it's something very cheap that anyone can do. Is actually you cross an existing, let's say, a local plant or an animal or a crop with some exotic one that you imported probably, right? So the definition is highlight, right? Page number 125. Plants and animals of different breeds or races are mated with each other. So in outbreeding, there are, of course, two parents. Two parents in one. The two parents are known by two nice names. There is a normal, let's say, local parent. We call it indigenous. Nothing special about him, right? So there's an indigenous parent. We are going to cross him or her with an exotic one. Exotic is probably from a different region or maybe a different imported one or something. So indigenous and exotic we are going to cross. Then this exotic parent probably has some nice qualities this indigenous one doesn't have. So the example we have given is what India, Zebu breeds of cows and nondescript cows are crossed with exotic breeds like Holstein Friesian, Brown Swiss and Jersey Bulls. ඔතන මේ Zebu කියලා කියන්නේ it's like I think it's something like the normal cows that you have in your road නිකන් පාරේ හැමතැනම ඉන්නේ normal. Nondescript කියන්නේ කවුද නිකන් you don't know the breed of them. Just a cow, right? Random cow. Those are Zebu or nondescript. Nondescript anything, nothing special about them, right? Just normal cows. You can cross them with Holstein, Friesian, Jersey Bulls, Brown Swiss. All those are imported varieties. And usually you don't need to bring the male. ඒගොල්ලන්ගේ sperm එක freeze කරලා you can import the sperm and you can cross. So what are you going to get? So you can get a nice generation progeny with increased maybe milk production, or they have more muscle to get more meat. This is outbreeding, right? Make, if you want, you can take down quickly, right? Small note, write down the topic outbreeding. Animals or plants of different breeds or races are mated together, that is outbreeding. Okay, still in genetics you will understand we are not, let's say, touching their DNA. We are not touching their DNA, not not yet, because we are going to touch DNA in today's lesson, molecular, right? We are going to take out DNA, we are going to put things additionally or remove things we don't want. DNA, yeah, changing by molecular techniques, but in genetics we don't usually do. What we are going to do is we are going to use normal reproductive system, let's say normal reproduction, but we are going to just control it to the way we want. right? So this outbreeding is a good example. The next one is even better, hybrid breeding. Okay. I'll erase this side. Hybrid breeding, very commonly questioned. You can get a short note also. Hybrid breeding. So pay attention. Online people, not sleep, right? Hybrid breeding, as the name says, what do you mean by hybrid? It has two power sources, right? So what we are going to do in hybrid breeding is we are going to combine two nice characters of the two parents, so you get one all-in-one progeny, all-in-one generation. So the definition is when genetically unrelated purebred plants or animals in the same species are mated with each other. Hybrid breeding. So to do this, we need some, let's say, preparation. එහෙම එක එකපාරට මොනවා I'm going to create a hybrid variety today කියලා you can't do. හිතිච්ච විදියට you can't create, you can't do hybrid breeding. It takes some preparation. What are the things you need to do before you do hybrid breeding? You need to have what we call as, let's say, purebred lines. Purebred, not farm. Okay, purebred lines. Or you need to have purebred lines of parents. How do you get purebred? How do you get purebred? You get purebred by only allowing inbreeding. What do you mean by inbreeding? I'll explain this with an example. Let's say, what is there any example given in your, let's say, මෙහෙම එකක් ගමු. So let's say you are going to make a hybrid rice variety. Rice. Tell me two things that is good to have in a rice variety. Maybe good round seeds on the right, big round seeds. Round seeds. Huh? Then another quality might be having many seeds in one stalk. Many seeds in one stalk. Again, if you take a, have you seen rice? Be karal kilan again. If you take a rice stalk, if you have a lot of seed there, that's a good variety, So if you have both in one, very good. So what do? You do what we did earlier, artificial selection. You choose a population producing very nice round seeds and you allow only them to reproduce again and again and again for many generations until you get a variety where everyone is making round seeds. Then in from another population you choose plants, paddy plants that are making a large number of seeds per stalk. Then you cross them among each other for several generations until everyone produces many seeds. Now you are ready to do hybridization because now what you have? You have two purebred lines. For over several generations, you have created paddy plants having these two qualities. So what you are going to do is you are going to combine and you are going to get your hybrid. This hybrid will have both in one. They have round seeds and they have many seeds in one stalk. Hybrid. Have you heard these farmers talking about hybrid yeah, not in our areas, but in agricultural areas, they have TV advertisements also. hybrid bija ganna අර මේවා කියලා hybrid seeds advertisements because they have more favorable qualities than normal plants because they are hybrid breed. They are bred in this way, right? So let's read the resource book. Generally, this is carried out with plants and animals who do not share common ancestors on either side up to 4 or 6 generations. Highlight. You have to create purebred lines for that long. The offspring of such a mating is known as outcross. මෙයා hybrid වගේම මෙයාට we can call him as the outcross. Because why? These are product of these inbreeding එකේ sort of outward product එක, right? Outcross. Offspring of, yeah, and they will possess stable characters and something very very important, hybrid vigor. Vigorous means what? They are more like, let's say, strong, right? Hybrid vigor සාමාන්‍යයෙන් very commonly you are asked in the SCQ paper, what is hybrid vigor? Have you seen questions? Okay, vigor, yes. Yeah, hybrid vigor, or you can call it what? Heterosis. Heterosis, right? You don't need make a onnang liyaga, you don't need this example, right? Write down the topic hybrid breeding. Write down the topic hybrid breeding. Okay, hybrid breeding. Then write down the definition. Genetically unrelated. Okay, genetically unrelated purebred plants and animals, genetically unrelated purebred plants or animals in the same species are mated with each other to produce an outcross. Outcross. To produce an outcross. This outcross will have, this outcross will have, this outcross will have, write down, this outcross will have hybrid vigor. This outcross will have hybrid vigor or heterosis. What is the definition of heterosis? Increase in characters such as increase in characters such as size, growth rate, fertility, yield. All these are increased in the hybrid organism because of hybrid vigor. They can ask you, what is hybrid vigor? What are the characters that you can see increased in the hybrid organism? Size, growth rate, fertility, yield. I'll erase this side. Now there's a problem with this. So you will think this is very good, no? Right? It's very good actually. But there's a problem. What would happen is, if you allow these hybrids and hybrids to cross mate, right? If you allow hybrids and hybrid to cross, that is going to cause a reduction in the hybrid vigor. Hybrid vigor is going to decrease, right? Decreased hybrid vigor. Okay, this is one of the disadvantages of hybrid breeding. Okay, so did you write down the definition of hybrid vigor? Okay, next point, write down. Hybrid vigor decreases, write down, hybrid vigor decreases, hybrid vigor decreases if the hybrids are mated together. Hybrid vigor decreases if the hybrids are mated together. So you can make a small comparison, advantages and disadvantages of hybrid breeding. What are the advantages? What are the advantages? There's three in your resource. There is yield improvement, extended growing season, quality improvement. Again, Manad, obviously, what do you mean by yield improvement? Definitely, yes. Hybrid produces more yield. Can be more rice or can be more quality rice or something. That comes under quality improvement as well, right? However, the yield is increased. Quality, right? Yield improvement. What do you mean by extended growing season? That means they you can have, let's say, make an apply when is for varieties that you harvest again and again. Nikang apples, so that you have the cultivation for a long time. Give me an example for a rambutan. What else? That you have the trees for a long time. I think rambutans are very hybrid. Yeah, they are. We do hybrid breeding rambutan. Mangoes? Mangoes. Yeah, no. Mangoes we do hybrid breeding. Yeah, for let's say trees, no, those kind of cultivations that you have, you cultivate again and again. If you have an extended growing season, that means the plant is producing fruit or whatever for a long period of time. That is something you can see in hybrids. They remain profitable for a long time. That's the meaning. Huh? Quality improvement means some things you expect from a fruit, no, some product. Maybe in examples, crispy taste of watermelon, right? Probably nice taste, no, right? Okay, some qualities you can improve in hybrids. Okay, those are given in the resource. Huh? There's three, I think. Next page, no. There are some disadvantages. What are the disadvantages of hybriding? Need many years of preparation. Many years of preparation. You need to prepare pure lines over a long period of time. Right? Also, it is expensive. Expensive because these hybrid breeders have to maintain pure lines over a long period of time. The seeds, or let's say the animals or whatever they produce, is expensive. Expensive, Ganavedi, because they are hybrids. Those are the two negative disadvantages. And also, if you cross hybrids and hybrids, the hybrid vigor decreases. You can also write it. If you cross hybrids, if you let hybrids interbreed, the hybrid vigor decreases. Those are the three disadvantages. Quickly take down. Hybrid breeding. Online people, any questions? What are the advantages? There's mainly three. You can say increase in productivity, right? You do a cultivation and you get more yield. That's the basic thing. Cultivation එකක්ම වෙන්න ඕනේ. It could be animals. Hybrid animals can produce more milk. Cows වැඩිපුර කිරි හදනවා. Or they are more, let's say, have more muscle to get beef. Chicken. Yes, that's also disadvantage. If you allow hybrids to interbreed, hybrid vigor decreases. You can write it down here. That's why, that's another, let's say, you need to maintain the parents continuous parental lines. That's why it's expensive. Every year, these big seed companies, they produce hybrid seeds. Why? How? They have the parent generations. So they every year they cross them, and they get seeds. That's why they sell them for high prices, because they are doing the work. They are maintaining pure lines. So that's another disadvantage. If you are like a, let's say, a poor farmer, you can't get hybrid seeds. Then his yields are going to go down, right? Okay, clear, no? We can Taggala, we can go to the final breeding technique. It's not a technique actually, Niga. Yeah, sometimes in the case of plants, it can be a technique. All clear, right? So you know now what I mean. All the MCQ points, theory points you can ask from here, right? HRI only, okay? Otherwise, because there are more important things to study in your syllabus, but the other questions are right. So we come to something interesting: interspecific breeding, interspecies. Interspecies breeding. As the name says, you are breeding two organisms of two different species. Okay. Interspecific between species. Right. So what's going to happen here? Write down the topic: interspecific breeding. Write down the topic: interspecific breeding. Okay, we are on page number 126. This is grade 13 resource, right? Grade 13 resource book. Online people, if you have the PDF, then fine. Molecular is in a separate book now, no, right? But in the PDF, it's all in one. If yeah, I mean the printed version, they have two books for grade 13, right? Yeah. In the PDF, you have grade 12 PDF, grade 13 PDF. That's yeah. Okay. Write down male and female organisms. Male and female organisms of two different species are mated. Male and female organisms of two different species are mated. So let's talk about this because this interspecific breeding works very differently. Did you write down? Male and female organisms of different species are mated. Huh? This works very differently in between animals and plants. Very, very different. Let's talk first about animals. In animals, it's not very successful. You can't cross between species animals. You can, but the progeny is going to suffer, right? You can only cross a few species. So, restricted to a few species. Huh? It makes sense, no? Right? If you even if you take mammals, so many differences. You can't cross maybe a monkey and a cat, right? So there are so many differences. No, even though they are animals, you can't cross. So sorry, then the mammals you can't cross. You have to have very very close relations. I mean, let's say so species. What is above taxon about that? Domain, kingdom, phylum, class, order, genus. Maybe common genus. At least you have to be. මතකයි නේද? Taxons මතකයි නේද? Online people, domain, kingdom, phylum, domain, kingdom, phylum, class, order, genus, familia, genus. I can't remember, that's why I'm asking. Genus, species, then sometimes subspecies. No? So we are crossing in different species. At least they have to be in the same genus. At least. Nathang it won't be successful. Right? You can't cross animals, different orders, different families, very unlike, right? So restricted to a few species. So some examples are given in your resource, right? Page number 126. යටම තියෙන්නේ. You have mules. Mules are by male donkey and a female horse. Hini, male horse and female donkey එක අනිත් පැත්ත. Right? Liger, male lion, female tiger. So these mules and hinnies, horses and donkeys. නේද? So they have a common genus that is called Equus. They are both of the same genus. So we can cross them. Shape එකේ. But they are going to have problems. We'll talk about the problems. Then liger, lion and tiger, no? They are both of the genus Panthera. Okay. Then there are something of the genus Equus, that is Zos. Zos and Zonky කියලා දෙන්නෙකුත් ඉන්නවා. Okay. Zonky and Zos. Zebras and donkeys, horses and zebras. ඒගොල්ලොත් ඔක්කොම Equus. Genus Equus. Okay. In your tute, I have given a photo of a liger. I have run out of tutes. Okay. Next week I'll give. Okay. Online people, in your tutes, I have given a diagram of a liger. Yes. Can you share? Yes. That's a liger. Lion. Samanen අහන්නේ නැහැ. Who is the father? Who is the mother? කියලා, right? Online people, diagram tute. I think page. Which page? So male lion and female tiger. Okay. Go home and Google and see how the zoos. So it's like a very large horse having like lavata stripes, right? Zebra stripes are sort of half there, half not. Okay. And mules very common, na? Mules ලට සිංහලෙන් කියනවා මොකද? කොටලුවා කියලා කියන්නේ. Right? They are stronger than a donkey and also easier to maintain than a horse. Stronger than a donkey, easier to maintain than a horse. Mules are very common. Okay. There's a big problem in animal interspecific breeding. What is the problem? You can explain it using this donkey and hold on, donkey and horse. So they say donkeys have how many chromosomes? 62 chromosomes. Horses have 64. Neither. So imagine, so you are going to make a mule. So you are going to take a male donkey, no? Male donkey again. A male donkey produces what? Sperm. Female horse produces what? Egg. Neither. So how many chromosomes are going to be there in the sperm produced by the donkey? In the sperm produced by the donkey, how many chromosomes? 31. Half, no? 31 chromosomes. In the egg produced by the horse, 32. So you are going to cross them and you are going to get a mule. This mule is going to have how many chromosomes? 63. Mule is going to have 63. Okay. So how is this mule going to have children? Imagine this is a male mule. He is going to produce sperm. Problem, no? How are you going to divide 63 by 2? Yes. Yes. Got it? And that's the problem. With animal interspecific breeding, usually they are infertile. infertile. Usually they are infertile. They cannot produce their own offspring. These ligers, hinnies, mules, they cannot have their own children. They don't produce functional gametes. Right? Okay. So this is the animal story. Okay. So divide the page into two. Right? Divide the page into two. Divide the page into two. Well, people, any questions? You can hear me clearly, right? You are not hearing another class like commerce like last time, no, right? Kalin sir, you learned some commerce, right? Yes. Next classroom we have commerce class. Okay. So divide it into animal, interspecific and plant. We have a lot more to write in plants. Huh? So first point, write down under animal breeding, animal interspecific breeding, divide it into two. Restricted to a few species. First point, write down. Restricted to a few species. Write down, write down. Okay. Restricted to a few species. Next, write down. Next point. Offspring can develop into adults. Offspring can develop into adults. Offspring can develop into adults. But may not develop. But may not develop functional gametes. But may not develop functional gametes. Within brackets, sterile. Sterile. වඳයි කියන්නේ sterile. If you want a reminder, write down the taxonomy on nang. Don't overestimate මතකයි කියලා, right? Write down the examples: mules, hinny, donkey, sorry, zonkey, zos. All those are genus Equus. Okay, mule, hinny, zonkey, zos are genus Equus. Liger is in the genus Panthera because lion and tiger then Panthera. And then write down the problem with donkey and horse. Donkey has 62, horse has 64. So when you have a mule, mule only has 63 chromosomes. තොර නිකන් 31 හමාරක් දෙන්න බෑනේ 31 and a half. එහෙම chromosomes නැහැ. So sorry, එහෙම sperms නැහැ. So it's going to be non-functional abnormal sperms. Right? Plant breeding වලට යමු. Plant interspecific breeding. Plant interspecific breeding. Plants actually tolerate interspecific breeding very nicely more than animals. Okay. they actually tolerate it very nicely. Why? It occurs naturally, no? Right? In plant, let's say reproduction, there's nothing controlling, right? Name them pollen from one plant, it goes in the wind and gets deposited on a flower, mehema, right? On a flower, what is this called? The stigma, right? On the stigma of a flower of the not the same species. May pollen එක ගිහිල්ලා කාලා get deposited on a different species flower. නේද? Because it's wind pollinated, you can't control it. So sometimes this can be successful. So usually plants tolerate and sometimes owner coming, it's a good thing. Interspecific breeding can be a good thing for plants sometimes, right? But it's more I mean, let's say it's always better than animal interspecific breeding because plants, you can say plant form is less stringently controlled. Again, what? The let's say the plant form. There's no specific structure to a plant exactly. If you take an animal, it has to have this number of limbs, this number of eyes, this amount of hair in his body, one tail. Right? So many fixed things. එක කකුලක් හරි වැඩි වෙච්ච ගමන් very abnormal, right? But if a plant has one more branch, more roots than normal, ඕනට වඩා පොඩ්ඩක් කොටයි නම්, it doesn't matter because plant forms are less stringently controlled. So it doesn't matter, few species get mixed up for plants most of the time, right? Plant form is less stringently happens. Natural interspecific breeding during pollination. You can ask, they can ask you an example for natural interspecific breeding. Pollination, no? The even a bee, Nemesek, he goes on lands on many flowers, no? එක එක ජාති මල් වල he lands. So he can take pollen from many different flowers to many different flowers. Interspecific breeding can happen. Okay, right. This problem is going to be still there. That the interspecific hybrid is sterile. The problem is still there. Sterile when a problem because why? The two species can have different chromosome numbers, right? So even in plants, the hybrid can be sterile, but plants are more. Let's say they have more tricks, right? The hybrid can be sterile, but sometimes, for example, they can undergo doubling of chromosomes and become fertile. For example, mm, yes, may example එකම ගත්තොත්, if you take the mule, if the mule can double his chromosomes, he is going to get 126. That is a number divisible by 2, no? Right? But he can't do that. But if a plant happens to have this sort of an odd number, what he can do, he can double the number of chromosomes and become fertile. Plants have those kind of tricks. The example given here is what? Highlight Page number 126. Bread wheats that humans use today are a result of two hybridizations, each followed by a chromosome doubling to produce fertile hexaploids. Hexaploids. Look at the example again. Right? The example for this bread wheat. Bread wheat. What has happened is so bread wheat is actually 6N. They have undergone doubling because they initially must have had 3, no? 3N. They underwent doubling to get 6N. Then okay, no? It's a number divided by 2. So plants can do that. So the example is bread wheat. Sometimes this new doubled fertile, let's say hybrid, can be the start of a new variety. Because why? He has more chromosomes, right? This can be start of a new variety. When you were studying this previously, did you go through this detail? Hmm. So this newly doubled, let's say hybrid, can be the start of a new variety. Some examples are given here. Um, boysenberries කියලා Knott's Berry Farm in California. It's a new variety of berries. Go home and Google. It's a famous company, yeah? American company. Knott's Berry Farm. They crossed blackberries, raspberries, and loganberries කියන තුන cross කළා. They came up with a new variety. Species cross කළා. Right? So these blackberries, raspberries are all different species. They have crossed them and got a new variety. Then another example, it's not the benefits are sort of endless. So you can get a new variety, and this new variety can have some more advantages, such as pest and disease resistance. Pest and disease resistance. They can have more, let's say, added advantages. The example given is winter hardiness of apple. Winter hardiness of apple. Winter hardiness means what? The apple tree is more, let's say, it can survive the winter season better than the normal apple because they have crossed one of them Malus domestica, Malus asiatica, Malus ඒව මතක තියාගන්න ඕනේ නැහැ. Okay, species නම් මතක තියාගන්න ඕනේ නැහැ. But remember, you can get this sort of new varieties by plant interspecific breeding, which is usually successful. Okay, write down under plant interspecific breeding. Plants hybridize more frequently and successfully than animals. Plants hybridize more frequently and successfully than animals. Plant form is less stringently controlled. May I? Stringent. Plant form is less stringently controlled. You can, if you like, you can write down this: natural interspecific breeding happens during pollination. Natural interspecific breeding happens during pollination. You can take this down. The hybrid can be sterile, but with doubling of chromosomes, it can become fertile. The fertile one can be a new variety which can have nice new qualities such as pest and disease resistance. Online people, any questions? So this week you are supposed to write the second paper. Last week I expected more answers for the paper class, right? In the paper class we are giving, I mean, we are ranking higher scorers, right? Top scorers will be sort of put in the group. Huh? So don't miss the opportunity. We are giving and we are sort of we are marking and giving you the papers. So send your papers if you have done the paper. Okay. Also, so those are the breeding techniques, right? Then we come to certain genetic breeding principles that we use in these techniques, right? You have three, that is polyploidy, mutation breeding, and genetic modification. Methanonym polyploidy is the most, the one you need to know in a lot of detail. Okay, right. Let's come to genetic techniques. I'm sorry, genetic principles. Huh? Genetic principles in these techniques. So we have learned what are the things that we can do with genetic knowledge. Now we are going to see what are the principles behind them. There's actually three in your syllabus. Ethering polyploidy is the one that you need to be thorough with. Okay. Right. Genetic principles. That means sort of the theory, no? Why how do you get this effect? What is the basis of it? what's the reason, right? So we have talked about hybrid vigor. When you cross, let's say, different plant varieties, you can get a, sorry, different plant species, you can get a new variety, right? We talked about some beneficial effects. How are they giving out beneficial effects? That's because of the underlying genetic principle. That's what we are going to talk about, right? So genetic principles in breeding. First one is the most important one, polyploidy. So we already talked about some polyploidy. අර bread එකක් ගැන කතා කරේ 6n bread එකක් ගැන. That's polyploid, right? Usually සාමාන්‍යයෙන් what is your ploidy? If you take one of your cells, මේ දැන් වගේ skin cell එකක් අරන් if you do a genetic examination, what is your ploidy level? 2n, no? දෙකයිනේ තියෙන්නේ, තාත්තෙන් ආපු කොපිය අම්මෙන් ආපු කොපිය to diploid, නේ? Right. But in polyploidy you are going to have more than two sets. That's the definition. Okay? Write down the topic. Genetic principles of breeding, right? We are making a short note, right? Genetic principles of breeding. First one, polyploidy. Write down the definition. Presence of more than two complete sets of homologous chromosomes per cell nucleus. Per cell nucleus. Presence of more than two complete sets of homologous chromosomes per cell nucleus. So we can do this by preventing certain cells from dividing mitotically, right? It's like this. Before mitosis, what's going to happen? So today, go home and read about the cell cycle. Na? What's happening before mitosis? What are the two parts of the cell cycle? You have interphase. Ne? Interphase කියලා එකක් තියෙනවා. Then you have M phase, no? Mitosis itself. නේද? These are basic knowledge. Interphase, what's going to happen? DNA replication. නේද? You do DNA replication. So imagine one of your human cells is going to divide by mitosis. It is initially what? 2n. Because of DNA replication, it's going to become what? 4n. After mitosis, it's going to come back to 2n. We are going to do, what we are going to do in polyploidy is stop this mitotic division from happening. So we are left with 4n cells. මෙතෙන්ට අපි ගහනවා anti-mitotic එක. Anti-mitotic එක. The example is given in your resource as colchicine. Colchicine. We can use this chemical to induce polyploidy in plants. Okay, so this is the principle. What are the things that you can see in polyploidy? Features of polyploids. There's something very important called Gigas effect. Gigas effect. එක කියලා එකක් තියෙනවා. Okay, Geiger's effect, Gigas, something. As the name says, what's going to do? Gigas effect means polyploids have larger organs. Larger organs. What are your larger plant organs? Leaves, roots, other tubers, fruits, flowers, or comma, they can be bigger in polyploids. Why? Because they have more DNA copies. They have more DNA copies, so their organs are going to be bigger, larger organs. Other advantage is what? Low growth rate. Flower later or over a long period. Polyploids. What is the benefit of flowering over a long period? That means the mal hadeno godak for a long duration. Let's say for a large duration of the year, the plant is producing flowers. This is good for a ornamental flower. Like rose variety එකකට වගේ. It's good to have a rose variety that is producing flowers most of the year. You can have it by having this polyploid. So this is an advantage, or better say let's call this advantages. First one is gigas. Second one is called genome redundancy. Genome redundancy කියන එක, it's an important concept. That means imagine in this diploid state we had a cancer allele, cancer causing allele. Okay, but now we have 4. When you have more copies or more DNA copies, it can, I mean, the sheer number can, let's say, mask the very few harmful ones. Because there are so many copies, the bad ones can become masked by the other copies. You get it? Because the DNA numbers, or you can say allele numbers, have double, triple, quadrupled. The bad alleles can be masked by the large number of good ones. So it's better to have more than 2n in 4n. So the masking effect is high in 4n. So that is genome redundancy. What is the definition of it? You can call it bad alleles or deleterious alleles masked by extra wild type alleles. Wild type කියන්නේ normal. හොඳ ඒවා. Wild type alleles. Sometimes there's something called functional diversification as well. That means these new copies can start to do a new function. Because you have so many extra copies, they can start to do something entirely new. Is species එක normally කරන්නේ නැති දෙයක්. Something that is not normally done by the species can start to happen because of the Gigas effect. Okay, finally, there is something called increased heterozygosity. There is also going to mask bad alleles and also increase the effects of good alleles. Okay. Then there are some disadvantages. Disadvantages. It can cause reduced fertility. Okay. For example, something something can become by polyploidy, they can reach a ploidy level that is අර වගේ odd number වෙන්න පුළුවන්. They can become an odd number because of the polyploidy that we are trying to make. So if it becomes an odd number like this, then fertility reduces again. Fertility reduces. They can't produce nice gametes. Odd numbers. Example is given in your resource, triploid watermelon. Right? Okay. Let's write down polyploidy. Did we write down the definition? Okay, write down. Polyploidy can be induced. Polyploidy can be induced by antimitotic agents. Polyploidy can be induced by antimitotic agents. Example, colchicine. If you want, you can write down that. In interphase, there is DNA replication. By stopping the mitosis, we are stopping at the double state. So we are ending up with double DNA sets. Then write down the advantages and disadvantages separately. Most important one, Gigas effect. They can ask you what is Gigas effect. What is Gigas effect? Polyploid individuals, Gigas effect write down. Polyploid individuals polyploid individuals exhibit larger organs compared to their diploid counterparts. Their diploid counterparts. මොනවාද සාමාන්‍ය organs? Leaves, roots, tubers, fruits, flowers ඔය ඔක්කොම ලොකු වෙන්න පුළුවන්. Write down the other advantages as well. Low growth rate, flower later, over a long period. මේ තුන, these three are especially beneficial for ornamental. What do you need? What do you think low growth rate is good? නිකන් ornamental පැලයක් ලස්සනට තියාගන්න එකක්, you have to keep it for a long time, no? එක විදියට. Right? So if it stays, if it grows very slowly, that's good. Right? ඔය නිකන් ඔය තියෙන මොනවාද lucky bamboo අරුමේව තියෙනවනේ, you plants you keep inside the house. If they grow very slowly, that's good, right? Ne? Certain ornamental species. Genome redundancy, they can ask you the definition. What is genome redundancy? ඉස්සරහින් definition එක ලියන්න. What is genome redundancy? Having additional gene copies. Having additional gene copies due to increased ploidy. Having additional gene copies due to increased ploidy. Having additional gene copies due to increased ploidy. So, what are the benefits of gene redundancy? Write down. It promotes a buffering effect. Chemistry වලට දන්නව නේද buffers? That means it prevents change. නේ? It promotes a buffering effect in which deleterious alleles or bad alleles are masked by extra wild type alleles. Are masked by extra wild type alleles. Then next one, functional diversification. Next one, functional diversification. Final advantage, increase heterozygosity. Right. Okay. Okay. So then we have mutation breeding and genetic modification, two more, and then artificial and natural breeding, disadvantages and advantages. That's it. We can move on to molecular after a break, right? Let's take a small break, 15 minutes. All right, people, come back at 3:20. Can you hear me, online people? Right. Let's quickly finish this. Quickly finish this. All clear about polyploidy, right? So we apply polyploidy only for plants, huh? Because you will see when you are doing molecular, if an animal has this sort of doubling, or let's say more than 2 copies, it's a massive mutation, chromosomal mutation. These sort of animals don't survive. They are even not even born, right? Not even born means actually at the sort of early blastocyst, that sort of very early mitotic cell division stages, it's going to get aborted, right? Nothing goes forward in animals, but in plants, as I told you, less stringently controlled. Genetic principles. Okay. Second one, mutation breeding, which is again what I just touched upon. Uh, so you can artificially induce mutations. Why do you need to do that? Why do you need to make mutations? Why do you need to make mutations artificially in order to breed? That's because what is the first prerequisite? What is the first thing you need to have in order to do some sort of artificial breeding? In genetics, what is the first thing that you need to, let's say, what is the first condition that you need to observe to decide I'm going to promote this? There has to be variation. Some sort of good and bad characters must be present within the population. If the entire population is exactly the same, how can you decide who are you choosing? You can't choose. First prerequisite is what? You have to have variation. That is the first prerequisite. So why do you do mutation breeding is when you don't have enough variation. That's why you do mutation breeding, right? So come towards the page. How many people you can hear me? Page number 128, mutation breeding. Why do you need to do mutation? Why do you need to induce mutation? Is when it appears there is little or no variability for the character to be improved available within the gene pool. Okay, no variability. Let's say you want a new bigger watermelon, but all the watermelons you see are of the same size. So how do you decide which one you? How do you choose to make the bigger one? You can't. You have to have bigger ones and smaller ones to see. I'm going to choose these people to cross and develop a new variety. When you don't have such variation, which is very rare, very rare phenomenon, mutation breeding is done very rarely. You will see why, right? That is why we induce mutation. We can induce mutations by two ways: by chemical agents, we call them mutagens, can be physical mutagens, right? Usually we call mutagens as carcinogens. Usually in a animal, these mutagens can almost always cause cancers, but in a plant we can usually use them to a benefit. Physical mutagens, ionizing radiation, ionizing radiation, alpha, beta particles, neutrons, gamma rays, protons. I think alpha is a proton. No, no, right? Alpha again होगा high helium 2 plus ion right? Beta again electrons. Beta waves again electrons, right? Okay. Then what are the other mutagens? Chemical. Few examples: sodium azide, ethyl methanosulfate. They can ask you this in MCQs. Sulfonate. They can ask you in MCQs. Then there are some important points, some disadvantages. Very difficult to do this. It's very difficult, or you can say limitations. There are so many limitations. First one, desirable mutations frequency is very low. If you produce 1,000 mutations, only one can be a good one. That's the meaning of 0.1, right? Out of 1,000 mutations, only one can be a good mutation, right? This is also in plants. In animals, almost all mutations are bad, deleterious, right? That's one problem. Then most mutations are recessive. How do you know this? Or fruit fly, Drosophila fruit fly, when they are mutated, their black and vestigial wings are recessive features. No, almost always mutations are recessive. That's a protective feature. When certain DNAs are damaged or certain genes are damaged, they the sort of the other parts of the animal's genome try to mask that, but making it recessive. Always, almost all mutations are sort of made recessive by the rest of the genome. It's a protective feature. So even when you induce a nice desirable mutation, there's a high chance it is recessive. So it is not visible. It is hidden by the dominant counterpart. That's another problem. Um, Usually limited usefulness in cross pollination. Limited usefulness in cross pollinated. ඒ කියන්නේ sexually reproducing plants. Cross pollinated. The thing is, when you are doing cross pollination, you are combining DNA from two plants, sperm and egg of two different plants. So, in such a situation, if you induce a mutation in one of the parents, there's a high chance again that mutation will be masked by the other parent's DNA. Cross pollination, hinda, right? So, usually they tend to be masked in cross pollinated plant species, usually only beneficial in self pollinated plants. That's another limitation. Yes, those are the limitations. There are some benefits that are given. We'll write them down. I don't need to write them on the board, right? Okay. So write down the topic mutation breeding. Did we write down the definition? Uh, write down the definition of mutation breeding. Online people, write down. Not asleep, right? Genetics. We are the very last part. Inducing desirable mutations. Write down the definition of mutation breeding. Inducing desirable mutations. Inducing desirable mutations in crop plants. See, it's only plants, huh? In crop plants using either chemical or physical agents. Using either physical or chemical agents when it appears there is little or no. When it appears there is little or no. When it appears there is little or no variability for the character to be improved. Variability for the character to be improved within the gene pool of the species. Within the gene pool of the species. Within the gene pool of the species. So nice long definition, but they can ask you structured essay, what is mutation breeding? Mutagens can be physical, can be chemical. Then write down the limitations. Desirable mutations are found at a very low frequency. The limitations, desirable mutations are found at a very low frequency. Then most mutations behave in a recessive manner, recessive fashion. Most mutations behave in a recessive fashion. Limited usefulness in cross-pollinated plants. Limited usefulness in cross-pollinated plants. Limited usefulness in cross-pollinated plants. Then at home you can write down the advantages. In your resource, page number 128, you have it has improved both morphological and physiological characteristics of crop and ornamental plants such as flower color, seed size, crop yield, disease resistance, salinity tolerance. Okay, write down the benefits of mutation breeding. Okay, drought tolerance, early maturity, wheat, barley, rice, potatoes, soybeans, onions. Okay, we have used mutation breeding. Okay, write down at home. Online people, you are home after the class. Okay. Finally, genetic modification, I'll leave it because that's something we are going to talk a lot in detail in molecular. So no need to talk about it right now. What we are going to do is directly we are going to modify the DNA itself. Okay. Here also there is some DNA interference, but when you are doing genetic modification, we are taking DNA from one organism and putting it to the genome of another. Direct copy paste करना, sometimes cut paste करना. So many different techniques are there in genetic engineering. So we are going to do that in molecule. Okay. Homework, I think last week also I gave homework. I'll send marking schemes for all these. Okay. Homework, write down. Homework, write down. Advantages and disadvantages of artificial and natural breeding. It's not a full essay. There's not enough points to be a full essay, right? Half an essay. Target for about 30 points. I'm not exactly sure, but I think about 30 points are there. Okay. Advantages and disadvantages of artificial and natural breeding. What do you mean by natural breeding? The breeding that is occurring if you go to a forest. Some sort of natural ecosystem, if you go, that is what's the breeding occurring there? Natural breeding. In the normal, sort of without human interference. Artificial breeding is whatever we do that we interfere with the reproduction of an animal or a plant or something that is artificial. So what are the advantages versus disadvantages? It's given in page number 129 and 130. I'll send marking schemes for these. I think last week also I gave some homework. Short notes dunna Hardy Weinberg, polyploidy. For all those, I'll send you marking schemes. Okay? Online people, did you write? I'll send you marking schemes. Okay? Okay, fine. Let's move on to this new topic, molecular biology and recombinant DNA techniques. Gradually, every year, the question weight from molecular is increasing. From 29, I mean, around 2019, 2020, the question weight was very low because it was just introduced. Even the teachers didn't know how to teach it. So many advanced techniques, advanced concepts were there in the molecular biology because sometimes these PCR, electrophoresis, those techniques are only found in, let's say, university laboratories, right? It is not, sometimes it's not even undergraduate level, right? PCR him, someone even undergraduates don't get to do, but they are in your A level syllabus, right? Because in our early years at medicine, we had biochemistry. We only learned about electrophoresis in university, but we already learned it in A levels. But even in university, we were not supposed to do electrophoresis or anything. Just what is the theory of it? PCRM, it's not even undergraduate level. There are some techniques called gene gun and what else? Transformations, yeah, so many techniques that are very advanced that is in your molecular syllabus. But what to do? You have to learn. Okay. Online people, any questions? So molecular has several subsections, several subsections that we have to tackle bit by bit. First, let's say subtopic is you have DNA packaging. Okay, that's what we are going to do today. DNA packaging, how DNA is packed into prokaryotic and eukaryotic cells, right? That's the first subtopic. Then we have DNA replication, how DNA is copied. Then we have the gene. What's a gene? Structure of gene, prokaryotic, eukaryotic genes, blah blah. Then we have gene gene expression. Gene expression protein synthesis. Gene. Then so 1, 2, 3. Then we have mutations. Then we have gene technology, gene techniques. So under this we have so many subtopics: DNA isolation, then restriction maps, DNA copying, as that means DNA cloning, restriction maps වගේ subtopics තියෙනවා, DNA libraries, what else? DNA fingerprinting, PCR. Finally, we have genetically modified organisms. Big, big lesson, na? Right? So this is the basic plan. We'll go through the lesson in much detail and let's say good enough speed. We have to revise, right? At the same time, we'll do the questions. The separate question tube, right? Okay. I'll send you all. So that I run out of tubes. Okay. Right. Right. So let's move on to the first. I mean, what we have to tackle today: DNA packaging. How DNA is nicely compressed into cells. How their DNA is found in cells has a very, let's say, massive influence on how DNA behave. How they are packed is very important in how they behave. Like cupboard එකේ ඇඳුම් තියෙන විදිහ අනුව it depends on how much time it takes for you to get dressed, how organized you are. Trip එකකට ලෑස්ති වෙන්න කොච්චර වෙලා යනවද, right? Depends on how organized, or let's say being organized and let's say if your clothes are very අහුමුළු වල දාලා තියෙනවා නම් it's going to make it difficult, but if you're organized that's going to make it easy. But again, if you're too loose, if your DNA is everywhere, if your clothes are everywhere, that's also a problem. If your clothes are very tightly packed and sort of compressed, that's also is going to make it difficult to get ready. If your clothes are everywhere, that is also going to make it difficult. You have to have a nice organization, no? A balance. That sort of story is there, how DNA is packaged or organized in different cells, mainly eukaryotic and prokaryotic cells. Okay, so in the first page, that is basically what we know about DNA from the first lesson, sorry, second lesson. Let's quickly go through it and highlight the things you want. Okay. um, Nucleic acids are capable in directing their own replication from their monomers. What are the monomers of nucleic acids? Nucleotides. Most organisms DNA is found as genetic material. Some viruses have RNA as genetic material, but are they organisms? No, viruses are not organisms. Okay. Accurate replication of DNA is transmission from one generation. Okay, right. DNA double helix. Who actually, let's say, discovered the structure of DNA? Rosalind Franklin. Highlight. By X-ray crystallography. It was Rosalind Franklin who discovered. She found out the model, and looking at that model, Watson and Crick proposed and described the DNA double helix. So you know all these things, no? Right? We have 4 types of nucleotides forming DNA, and they are arranged in a double helix, spiral staircase with it. Right? Online people, there's a spiral staircase here. So what are the handrails? I guess high handrails දෙකක් තියෙනවා. Can you see? It's too bright. What are the handrails indicating? Backbone. Sugar, phosphate, backbone. What are the steps? Nucleotides. Deoxyribonucleotides. Right? What are the two types of deoxyribonucleotides? You have purines and pyrimidines, and always a purine binds with a pyrimidine according to a base pair rule. Adenine always binds with thymine by 2 hydrogen bonds. Guanine always binds with cytosine by 3 hydrogen bonds. Simple law. That's all is there in the first page, right? Right. Still on page number 1, architecture of chromosomes. Okay. Highlight the definition. What is architecture of chromosomes? What is architecture of chromosomes? Very sorry. There's something you need to highlight above. T.H. Morgan ke කෙනෙක් ඉන්නවා. T.H. Morgan, they can ask you in this MCQ sometimes. Highlight. T.H. Morgan, through their experiments, concluded that chromosomes are made of DNA and proteins. Chromosomes. How do you get chromosomes? So there's this histone killer proteins. No? There's a type of protein called histones, and when the DNA double helix, this is the double helix, when it wraps around this histone, 8 histones twice, you get what? Chromatin. Eh? We are going to talk about this. We are going to get chromatin. Chromatin condenses to chromosome. So what did T.H. Morgan said? Chromosomes, ඔබලා resource වල තියෙන chromosomes කියලා. Chromosomes, okay? Chromosomes are made up of DNA and protein, and genes are regions of chromosomes. A gene is a section of chromosome. That's it, basically. Okay. So who said that? T.H. Morgan. Rosalind Franklin, Watson and Crick, T.H. Morgan. ඔය ටික තමයි history එකේ ඕන අය. Okay? History of DNA. Right. We were talking about architecture. Highlight the definition of architecture. Architecture. There are two possible ways DNA can be arranged. What is the architecture of chromosomes? Is the way DNA molecules are arranged in the nucleoid or nuclear region of the cytoplasm of prokaryotic cells or in the nucleus of eukaryotic cells. So if you take prokaryotic cell, where do you have DNA? In the nucleoid, or you can say nuclear region. You have to specify, yeah. If it's eukaryotic, where do you find nucleus? Okay, right. Then next page, DNA of both prokaryotes and eukaryotes are called chromosomes. Next, very very important. However, true chromosomes are present only in eukaryotes. Highlight. True chromosomes are present only in eukaryotes. Why? Why true chromosomes only found in eukaryotes? Histones associated with DNA, is it present in prokaryotes or no? Second lesson. Ne? Second lesson, prokaryote eukaryote cell comparison. Right? Size, DNA, cell wall structure. What else? Nucleus structure. Eukaryote DNA, eukaryote prokaryote comparison. Structure of flagella. Presence of organelles. What else? Type of ribosomes. Right? Go home and go through that. Okay, second lesson. So, do you have true chromosomes in prokaryotes? No. Because why? There is no histones associated. There are other differences you will find out as we go along the lesson. Okay. Prokaryotic bacterial chromosome is a single double-stranded circular DNA molecule associated with few proteins. Eukaryotes have several chromosomes, each consisting of a single double-stranded DNA associated with histones and other proteins. With all the size of with the size of all chromosomes in an organism is considered, it's an enormous amount of DNA. Cell has a huge problem in having its DNA either in the nucleoid or nucleus. Right. Then highlight the definition. Containment of the genome or the DNA. Found out the place, right? Containment of genome or DNA in the nucleoid or the nucleus is called DNA packaging. Page number 2, above the topic architecture of prokaryotic chromosomes. Containment of the genome or DNA in the nucleoid or in the nucleus is called DNA packaging. Right? Why do we need to package? Because it's an enormous amount of DNA. Nicely pack කරන්න ඕනේ. ඇඳුම් ගොඩක් තියෙනවා වගේ. When you have a lot of clothes, you need to nicely organize them. Otherwise, මුකුත් හොයාගන්න හම්බෙන්නේ. Right? Clear. So we are going to move on to prokaryotic chromosomes. Mm, Shall we write down some things? Right? Shall we write down some things? Right? Under molecular biology and recombinant DNA technology. Okay. Let's make a small note. Subtopic architecture of chromosomes. Subtopic architecture of chromosomes. Architecture of chromosomes. Architecture of chromosomes. What is architecture of chromosomes? The way DNA molecules are arranged in the nucleoid or nuclear region of prokaryotic or the nucleus of eukaryotic. Write down that. Then next definition, DNA packaging. DNA packaging. DNA packaging. Write down the definition of DNA packaging. Containment of the genome. Containment of the genome. Containment of the genome in the nucleoid or the nucleus. Containment of the genome in the nucleoid or the nucleus is DNA packaging. In the nucleoid or nucleus is DNA packaging. Okay. Right. Subtopic architecture of prokaryotic chromosomes. How many chromosomes do prokaryotes have? If you take a prokaryotic cell, how many chromosomes would a prokaryotic cell have? How many chromosomes would a prokaryotic cell have? There's only one. There's only one usually. It's what we call as a circular chromosome. Okay. Very simple arrangement. There's an unfolded circular chromosome like this. Okay. This is circular. Level 2 naya chromosomes packed in prokaryotic cells only in 3 steps. Saraniga shirt you how do you fold a shirt? I don't know. Right? You fold the arms, then you fold the two sides, and then you fold three, right? How prokaryotic chromosomes are only packaged in 3 steps. 1, 2, 3 pack. Right? Circular, unfolded. This is after you take them out of the washing machine or whatever. Circular, unfolded chromosome. Average size 350 micrometers. Length 350 micro. Is it given? Yes. Then what's going to happen is there's a nice RNA core that is going to help, right? RNA protein mix that is going to help to pack this. ඒක නිකන් අර මේ how do you it's like අර නූල් බෝලයක් ඔතනකොට මැදින් මොකක් හරි දාන්නේ, right? When you are sort of organizing a ball of string, sometimes you put like a piece, like a tube in the center. So this is going to help to nicely organize this circular chromosome. So it's going to be attached like this. This circular chromosome is going to be attached like this, to this RNA protein core. Okay, online people, can you see this RNA protein core? It's a core, right? That is going to shrink its size significantly to about 30 micrometers. 30 micrometers. Final folding, what's going to happen is, I'll draw it here. These curves around this RNA protein core, these loops, these loops are going to further coil tightly, to the final nicely condensed level. There is only about, I think, 2 micrometers. Huh? These are average sizes. Online people, can you see that corner? Yeah, no? Is it glary, no? Right. This is how eukaryotic chromosomes are packaged. Let's quickly read. Packaging of DNA in prokaryotes is facilitated by proteins associated with the DNA molecule. Proteins cause the DNA to coil and supercoil so that uh, the DNA molecule is compacted into the nucleoid. Prokaryotic have a there's no nucleus, only a region. DNA molecule initially coil into loops, and these loops independently supercoil into domains. So මේකට අපි කියනවා this step we call this coiling. This step we call it supercoiling, and these are called loops. These loops, once they have supercoiled, they are called domains. Okay, and then write the subtopic prokaryotic chromosomes. Quickly write this, draw this with the sizes, 350, 32, 350 micrometers, nothing more product effective than you making your own note, right? Active learning, because you already have a note resource book, right? There is no use in writing the same thing again and again and again on different pieces of paper, right? Have a rough idea first, then study the resource. these loops there, it can change from bacterial species to bacterial species. This is like a average, right? What the typically happens, typical model, right? So you know what's the usual range of prokaryotic cells? It can be zero point 0.5 to 5 micrometers, right? So sometimes they can have less DNA. That means they can have small this sort of supercoils, right? So 2 micrometers is still within the largest bacteria size. So this is very close to a large bacterium, right? What is the average range of sizes? 0.5 micrometers to 5 micrometers. Prokaryote size. Huh? What are the three levels? This is circular unfolded. This you have to name. Huh? This is folded. This is folded. This is supercoiled. Huh? That is circular unfolded. This is folded. This is supercoiled. Okay, quickly write down underneath. Still drawing. Online people still drawing. Okay. Then write down underneath this. Write down underneath this. Functions of functions of RNA protein core. යටින් ලියන්න. Functions of the RNA protein core. These are questioned in your structure disease. Right? Functions of the RNA protein core. What is this core? Core. Maybe? Right. මෙතන තියෙන. Okay. RNA protein core. What are the functions? Individual domains. Write down. First function. Individual domains. What are domains? These loops. Huh? These loops. Supercoil. We call them domains. Right. First function of the core. Write down. Individual domains can relax and supercoil independently. Individual domains can relax and supercoil independent. Individual domains can relax and supercoil independently because of the core. Okay. Second function, write down. Core acts as a barrier for rotation. Core acts as a barrier for rotation by attaching the chromosome by attaching the chromosome by attaching the chromosome to the membrane. by attaching the chromosome to the membrane. Okay. So when these individual loops are, let's say මෙහෙම, right? So this is how your cupboard is packaged කියලා හිතන්නකො. So your these individual loops have different let's say functions of the bacteria. හිතන්න this is a bacterium chromosome. There are information related to their replication, related to their sort of protein production, toxin production, maybe their movement, maybe their respiratory reactions. So many genes are there. No? So imagine the bacteria is going to make a toxin, right? It's going to make a toxin. Then what is it going to do? It's going to relax one of these domains, read the gene, then again supercoil it. And you take the shirt you want and then you put the rest of it back. It's all organized into a nice cupboard, right? You take what you want, read it, and then you put it back. That is possible because the RNA protein core is holding everything together. Got it? Very commonly questioned section. Okay, clear? This is all you need to know. Other than that, there's a small point mentioned in your resource, page number 2. මේ diagrams වලට යටින් බලන්න. In addition to the chromosomal DNA, certain prokaryotes have extra chromosomal DNA elements. Extra chromosomal DNA elements called plasmids. Highlight plasmids. Highlight the word plasmids. These plasmids are going to be very important in upcoming DNA techniques. Okay, upcoming DNA techniques. These plasmids are going to play a massive role. Massive role. Okay, right. That's all about prokaryotic chromosome packaging. Simple, very simple. Any questions? What happens? There's the single chromosome. It is circular and unfolded very big. Someone cell එකක් වගේ 100 times either big, right? Very unrealistic. No? So what you are going to do? You are going to pack it in 2 steps. You are going to coil it, supercoil it. Coiling attaches it to RNA protein core, forming loops. The loops are going to be supercoiled into domains. Domains can be individually relaxed, read, and again supercoiled. Clear? Now we can move on to eukaryotic DNA package. Very important. There are, it's very similar and also slightly more complicated. That's the only thing, right? Make a steps. It has 5 steps. Yes, 5 steps. Okay. Eukaryotic DNA packaging has few more steps. What are the other differences? We have a large number of chromosomes. No? If you take the human genome, how many do we have? 46. Now, a donkey 62. So that means the donkey is more complicated than us. Right? Not really. The number of chromosomes you have doesn't mean they are complicated forms. Right? Eukaryotic chromosome package. Right? So, what are the main differences one thing, we have a large number of DNA in a eukaryotic cell, and those large number of DNA form usually multiple, right? A lot of individual chromosomes. So those individual chromosomes have to remain individual, right? මෙයාගේ chromosome number 1 ගේව chromosome number 18 වලට යන්න බෑ, 18 ගේව 15 ට එන්න බෑ. එහෙම mix වෙන්න බෑ, right? That's going to change how they are read. So this independence of the chromosomes have to be maintained all the time. That's one thing. And the other thing is there's a lot of DNA to be packed. So when you are considering those things, eukaryotes need much more complicated way of packaging DNA. Okay, right. So first thing is, what we need to do is we need to take our double helix and let's make chromatin. We මුලින්ම chromatin හදමු. So we are going to take our double helix like this, and so it's getting smaller. Okay, this is the double helix, right? I'll take it to one sort of string, right? I'll take it to one sort of compress it to one string. Then what are we going to do? We are going to wrap it around histones, histone protein. How many histone proteins? 8. මෙන්න මෙතන 4යි, තව ඉවසෙන් තව 4කුත් තියෙනවා. 8 histone proteins around. You're going to wrap the DNA. How many times are you wrapping it? Once and twice. 2 times. මෙහෙම ඊළඟට this DNA is going to come again, and we are going to wrap it around 8 more histones to produce another unit like this. What is this called? These are called nucleosomes. So this DNA molecule is going to continue like this. What is this level? This is what we call as first level, or chromatin. Well, use a different color. Right? This is chromatin. What is the usual size of a chromatin fiber? 10 nanometers. 10 nanometers chromatin size. Okay. Then what's going to happen is, so these, what are, give the names to this? What are these individual things? These are nucleosomes. Nucleosomes. Okay, level 1 done. There's a name we give for these segments of DNA that is in between two nucleosomes. Anyone? The DNA segments that link two nucleosomes are called, on the creative number then, linked DNA, right? Linked DNA. Easy to remember, right? Linked DNA. Second level, level number 1. Then it's very difficult to draw this, right? So level 2, what's going to happen is, these histones are going to sort of form a fiber. I'll sort of draw it like this, right? They are going to coil like this and form what we call as a 30 nanometer fiber. So now can you see, we are prokaryotic, what we did was, we are going to make it smaller. What now we are going to do is, we are going to make it sort of smaller size. We are going to get a lot together and make it bigger. It's like taking a single string and winding it around like a, let's say, නූල් බෝලයක් හදද්දි, sort of that නූල් බෝලේ becomes bigger, right? Because that's what we are doing in eukaryotic. We have so much DNA. We have to take all that DNA and step by step we have to nicely pack it. So we come to the second level. This is called 30 nanometer fiber. So slightly bigger. Level 2. This is level 1. This is level 2. third level, what's going to happen is this fiber is going to be arranged on a protein scaffold. What do you mean by protein scaffold? It's like a supportive structure that is going to act as like a basement, right? Like a flow එකක් වගේ, right? So this 30 nanometer fiber is going to be arranged like this here. And if you zoom in, actually this 30 is මෙහෙම තියෙන්නේ, okay? Right? Actually it's like this, but we are drawing it like this because we are zooming out. We can't see these coils now. You understand? So we jump a massive size, 300 nanometers, level 3. Then what's going to happen is this protein scaffold is going to fold. මෙහෙම ඒක fold වෙනවා. Right? Then with that, the DNA on it is going to fold as well. This is level 4, 700 nanometers. Level IV. Finally, this is going to form our final chromosome. That is what we call as අර වර්ණ දේහ කියලා කියන්නේ in Sinhala. Because why? This structure is visible by electron microscope, 300 nanometers, right? At this level, it becomes visible by electron microscope, right? Level 5. You can see it, it went back to a certain condensation, no? The 700 in the light went back down a bit in size because this is like the final level, right? So in here you will see that it's so tightly packed, tightly packed, right? At the final level. So these are the 4 levels. Let's read the resource and then let's draw this. Page number 3. Page number 3. Eukaryotic chromosomes are associated with a large number of proteins called histones. Highlight histones. Histones. That helps to organize DNA inside the nucleus. This DNA protein complex is known as chromatin. Highlight what is chromatin. It's a DNA histone complex. Okay, highlight. This chromatin, I'll talk about this again, right? This chromatin can be tightly packed as euchromat. Lightly, lightly packed as euchromatin or tightly packed as heterochromatin. Think of it like this. The clothes you wear every day, you don't pack them very tightly, right? Like all the everyday your denims and t-shirts, you don't pack them very tightly, right? The clothes you wear every day, your school uniforms or whatever, you sometimes your school uniforms, you never put them inside, right? Because they are frequently getting used. But if you come to your, let's say, formal shirts, formal things, where do you keep them? You keep nicely iron them and put them deepest part of your cupboard because you don't use them very frequent. So it's like this. Some of your DNA is used very frequent, right? Some of the DNA is not used at all or very rarely. So it makes sense that the DNA used very frequently is not very tightly packed. The DNA used very frequently, used less frequently is very, what did I say? DNA used very frequently is loosely packed. DNA used less frequently is tightly packed. Okay. What are the features of this euchromatin? Euchromatin, they are rich in genes. Rich in genes. Active in transcription. What is transcription? What is transcription? DNA දිහා බලන් mRNA හදන එක. Yes, transcription. DNA එක දිහා බලන් mRNA making. This mRNA දිහා බලන්න we are making proteins. Remember, right? We'll come there. Okay, protein synthesis, this whole process. This is how we make a protein, right? By looking at the DNA, we are going to make an mRNA. That mRNA is going to come out of the nucleus and go to the ribosome and make proteins. So usually euchromatin is active in transcription, but heterochromatin is usually inactive. Heterochromatin is usually inactive. They may regulate, sorry, they may involve in gene regulation. They ඒගොල්ලෝ actively proteins හදන්නේ නැහැ, but they are regulating the everything that is happening. ඒගොල්ලෝ නිකන් අර silently ඉඳගෙන they are controlling, right? There are things called epigenetic inheritance. You know these things, right? What is epigenetic inheritance? Inheritance of certain features that are not controlled by DNA information. What do you think about that? So we always think that everything that you see in a person, if you take a person's appearance and their behavior, you think everything was determined by their DNA. Not really, right? The person who's here, or you, all of you, look this way, behave this way because of two things: because of your DNA genetics, yes, and then your environmental factors, right? But in your genetics, other than the information in your DNA, that is your adenine, thymine, guanine, cytosine, ඔය ටික අමතරව there are some factors that can determine how your DNA is expressed. That is called epigenetics. That means outside genetics. So for example, if you have this sort of a gene, මෙහෙම gene එකක් තියෙනවා, right? It's adenine, thymine, guanine, or cytosine, guanine. Or විදිහට I again thymine, adenine, like that DNA is there. Suddenly there's a methyl group that is bonding to this DNA. This methyl group is going to mask this DNA. So that is something coming under epigenetic inheritance. So this part of DNA is going to be masked. So the DNA information is there, but it is not being expressed. That is called epigenetic inheritance. So such things can be controlled by heterochromatin. And finally, there is something called chromosomal integrity. Right? Integrity. That means they are sort of related to their stability. Yeah? That means they are sort of less likely to change very frequently. Heterochromatin කියන එක්කෙනා, because it's very tightly packed, he doesn't allow DNA to change. Chromosomal integrity. So those are the features of euchromatin and heterochromatin. Then we come to the levels. First level, double helix winds around histones. Then they are called nucleosomes. Nucleosomes are interconnected by linker DNA. This appearance is called beads on a string. Okay, beads can one the pabalu. It looks like a necklace, right? Second level, they twist in a spiral fashion to form a chromatin fiber of 30 nanometer size. Then it arranges itself on a protein scaffold. This black color is called a protein scaffold. It's like a basement support. Neither, right? Protein scaffold. That is the size of 300 nanometers. That is going to this scaffold is going to fold. Scaffold like a fold, 700. That is going to twist again to form your 300 nanometer. What is this? If you double it, right? If you draw the other sister chromatid, right? If you draw the other sister chromatid, මෙහෙම connected at their centromere, you can call this what? I mean, double line. It's about to divide. Mitotic chromosome. Okay, I'll ask you a question. Is DNA packaged into chromosomes always? If you take a cell from your body right now, is it packaged into a chromosome right now? A normal cell. Is it packaged into chromosomes? No. It exists as chromatin. What is the only situation in eukaryotic cells? DNA is packaged into this. About to divide. When it is about to divide in the interface, after DNA is double, it's going to undergo packaging to form chromatin. Matakai? What is the phase? Prophase 1. Sorry, I mean prophase. In prophase, what's going to happen? DNA, you can say DNA undergo condensation. It's the same thing, right? Normally, in a non-dividing cell, how is DNA as chromatic? When it is about to divide, it undergoes all these to form chromosomes. Okay, Taggala draw this. Quickly draw this. Divide chromatin into euchromatin and heterochromatin. Divide chromatin into euchromatin and heterochromatin. Write down their features. Write down their features and draw the 5 levels of DNA packaging in eukaryotes. Online people, I'll send a diagram to the group if you want, but I think this is more clearer than the one in your resource, which is a bit confusing. Any questions, online people? Okay, done. All right, people, any questions? Yeah, yeah. Hold on. Which one? Ah, yeah, no. Yeah, yeah, yeah. Yeah, I check one of my notes as well. Yeah. Um, so in the new resource, are there different sizes? This is supposed to be on the protein scaffold. It's 300. What is the size given for this level 4? Hmm? Is there a different value? 300 for the, okay, 700 says 300, right? Yeah, it has to be corrected, no? Yeah, 700. Yeah, yeah. So this is individually, yeah, it's given in the new resource or it's a mistake that needs to be corrected. Yes, yes. I checked one of my lecture notes as well. Yeah. One arm is 700. One arm is 700. So both arms come to 1400. Okay. Yes. Thank you. Yeah. Yeah. 10 nanometer, 30 nanometer, 300, 700, 1400 with the two arms. एक arm में 700. Yes, correct it. Okay. Right. Let's write some points. Right. Let's write some points. Let's write down some points underneath. Okay. In non-dividing cells, in non-dividing cells, in non-dividing cells, DNA exists as chromatin. In non-dividing eukaryotic cells, DNA exists as chromatin. In non-dividing eukaryotic cells, DNA exists as chromatin. Which is of two types, as euchromatin and exists as chromatin, which is of two types, as euchromatin and heterochromatin. So you wrote down the two functions, right? Different roles. DNA undergo packaging, or you can say chromatin undergoes packaging. Chromatin undergoes packaging or condensation, where in the prophase of mitosis. Those are basic concepts that tend to slip out. Huh? Okay, right. So, yes, so next week we can start DNA replication, and I'm going to finish DNA replication and then start the gene structure and also, if I can, gene expression. Then so DNA replication is a very likely essay topic, and protein synthesis are very likely essay topics, right? As soon as possible, we'll go there because we have to spend some time on gene technologies, right? Those are complicated concepts. We'll do questions. I'll send you the right. I'll next week give you the question tube, right? Separate question tube is there, and yes, online people, your delivery will happen within this week for sure, including the flash recaps and everything. Okay. Yeah. Any questions you want to ask? Anything? So, if you are joining the paper class, paper discussions happen on Thursday. We are doing our first full paper these days. We had the MCQ discussion last Thursday. This Thursday, I won't be doing the class. I have an exam. But I'll cover that discussion as an extra session later within this month. Um, so we have to discuss the part two, SEQs and essays. For the essays, I'm giving marking schemes, and the SEQ paper I'm going to discuss in TOEFL, right? So it was a paper only from grade 12. I'm going to do only grade 12 papers until the end of February. Then we are going to start grade 13 gradually. And in the last 2 months, we are going to do probably 3 full papers, full model papers. And at the same time, from February, I'm starting a past paper discussion online program, right? 2015 onwards, past papers online. Okay, that's going to happen on a weekday late night. Whacker. And there's another program that is part of the, let's say, paper class. Guest essay. Guest essay sessions කියලා session එකකුත් යනවා. Right? Every week we are going to discuss a guest essay after the past paper discussion. That's all online, nah? Right? The only physical things happening are this revision class and the paper discussion. Okay? Paper discussion and the revision class are physical and online. PO online, I'm going to do past paper discussion, essay, guest essay discussions. That's it. So you don't have to do past papers by your own. What you have to do is just keep your self studying, keep doing your self studying. That's it. Okay. Past papers I'm going to do from 2015 onwards. I plan to do 2015 onwards. Let's see. Okay. Because all these older syllabus papers, you can't do around 20% of the content. Some things are out of syllabus. So I'm going to remove those questions and we can still do. Right. There's only 6 papers, 2019 in the 2025 and come. So those are a few of the plans. Okay, February onwards, we are going to start massively, right? Full on focused program, right? Okay, right. Okay, we can finish for the day. Online people, any doubts? This next Sunday, I'll let you know for sure. Okay, if I am giving a holiday, everyone is in the group, right? Yes, in the group. I'll confirm in the group. If I am canceling, I'll have an extra online session that you all can join. Okay, at a convenient time, late night. Okay, cover up අනිවාර්යෙන් කරනවා because I have this 6-month plan to revise everything. Right? Okay. Okay, you're welcome. Right. You can. Class number do you have? You can send it to that number as a team. Then you can send that. Yeah. You also, you can either stay and I gave them the copy. You can either stay and write the paper. Yeah, sure. From a percent, then you can stay here and write the paper and give to the office, or you can go home and send the paper. I know you are staying and writing. But, okay.